So if we take this polygenic inheritance, many genes exhibit only character with many traits. So when we see the NCERT definition, when a character is exhibited by 3 or more genes, this is known as polygenic inheritance. So poly means many, genes means genes. When many genes combinedly, they are just exhibiting or governing only one character with many traits, that is called as polygenic inheritance. And we can see that there are two examples we can see, that human skin color and the human heights. If we take this human skin color, this is most important example for NEET point of view. This will become your PYQ for NEET exam. This polygenic inheritance, which is the most important example for it, it is this human skin color. This human skin color, it can be affected by environment. When it is infected by environment, it cannot be inherited. It can be inherited. But if we see, when it is affected, this human skin color is affected by the genetic control, or it is under genetic control. In genetic control, 3 genes are responsible for this human skin color. That is A, B and C. These genes are named as A, B, C. These 3 genes, these are just affecting this human skin color. In polygenic trait, we can say the phenotype. If we see the human skin color, there are many colors, but color is only one character. In this one character, there are many traits. That all traits are exhibited by these 3 genes. How these 3 genes? Because there is a dependence of the dominant and the recessive alleles in these forms, they will exhibit different traits. So we can say in this trait, when there is a very dark skin color, it will have all the alleles. So in these 3 genes, there will be maximum 6 alleles. When 6 alleles are dominant, then it will produce a dark skin color. When all the alleles from this ABC are recessive, like small a, small a, small b, small b, small c, they will exhibit a very light color, or we can say white color. Is this clear? So if we count these allelic dominant forms, or if we count the allelic recessive forms, each allele is the contribution of each allele. There is an effect onto this skin color. We can say that the phenotype reflects the contribution of each allele. That is, the effect of each allele is additive. When there are more dominant alleles, the color will be more dark. When there are more recessive alleles in these genes, the color will be very light. So we will see this in the cross. Okay, this is clear. Now, have the cross. Let me first rub the board. Assume that the human skin color is affected by 3 genes, that is A, B, C. So, assume that human skin color is affected by 3 genes, that is capital A, capital B and capital C. So their dominant forms, if we see, dominant forms capital A, capital B, capital C, they are responsible for dark color. These are responsible for, or we can see like that, the dominant form, let us have a cross between capital A, capital B, capital C, it is crossed with small a, small b, small b, small c, small c. When all the alleles are dominant, they will affect the skin color and it will produce the dark skin color, dark color, or we can say very dark color, and this will produce very light color. As we mentioned here, in a polygenic trait, phenotype reflects the contribution of each allele, that is, the effect of each allele is additive. If we see, these two alleles are dominant, these two alleles are dominant, these two alleles are dominant. If we see all the six alleles are dominant, they have produced very dark color. But in the same gene, when we are having all the six alleles recessive, they will form the light color. If we cross these two parents, this is parent 1 and this will be the parent 2. When we cross such parents, the gametes will be formed. It will form a similar kind of gamete. It will form a similar kind of gamete because there is no heterozygous form. These are homozygous parent, this is homozygous parent, and they will form a similar kind of gamete. That will be capital A, capital B, capital C, this will be the gamete. And here similarly, this will be small a, small b, and small b. This will be the gamete. And these are gametes. Let me write it here. These are gametes. In these gametes, when there is a fertilization, or we can say the hybridization, then you will find the F1 generation. In F1 generation, you will find this is capital A, small a, capital B, small b, and capital C, small c. This will not be the dark, this will not be the very light. So, in Mendel's monohybrid cross or dihybrid cross, the F1 generation was resembling to one of its dominant parent. But here what we can see that this type of progeny, it will not resemble to both of the parents, but the color of the skin will be intermediate. Is this clear? So when you are crossing this, then you will find the intermediate color. It's an intermediate skin color. So it will have intermediate skin color. Intermediate skin color. If we see, there are three dominant alleles, 1, 2, 3, and there are three recessive alleles. So the allelic forms, the number of dominant alleles and the number of recessive alleles, it varies. In parent it was all 6 dominant alleles, in this parent it was all 6 recessive alleles. But in where, if we see in the F1 progeny, when we crossed these two parents, we found that there are 3 dominant alleles and 3 recessive alleles. They have produced this intermediate skin color. Is this clear? Such inheritance is known as this polygenic inheritance. If we see this statement again, the phenotype reflects the contribution of each allele. So each allele, when dominant, it is produced very dark color. Each allele, when recessive, this has produced light color. But here is the difference in the number of dominant and number of recessive alleles. Only 3 dominant and only 3 recessive, they have produced this type of color. When we take it as self cross, thus यहाँ पर हम क्या देख सकते हैं? Let we see thus number of each type of allele in the genotype determines the darkness or lightness of the color. This inheritance also can be called as quantitative inheritance. So we can see this polygenic inheritance is also known as quantitative inheritance, in which we are just assuming the number of the dominant allele and number of the recessive allele. On to the basis of the number of dominant alleles or number of recessive alleles found in an organism, it will exhibit its own character. So we can say that polygenic inheritance is also known as inheritance is also known as quantitative inheritance. you will find also or determine, we can determine also the number of phenotypes in F2 generation. On to the basis of number of alleles, it will be equal to 2n plus 1, where n is equal to number of genes. So if we are having 3 genes, then 2 3s are 6 plus 1 in terms of human skin color. So in human skin color in F2 generation, we will have number of phenotypes is equal to 7. So let us have the cross, that is the self cross. In self cross, we will find out Dear students, we will see the selfing of this cross. So, in the F1 generation, we have the progeny, it's F1 generation. The progeny was capital A, small a, capital B, small b, capital C, small c. When we take the selfing of this F1 generation, then you know that selfing means self cross. It will be capital A, small a, capital B, small b, capital C, small c is crossed with capital A, small a, capital B, small b, capital C, small c. This is the selfing. Now, let us form the gametes. Gamete formation is very difficult here, but I will tell you the formula how to determine the gametes. First, we will determine the gametes by the way of formula, utilizing formula. So, how to determine? Determination of gametes. We know the formula. It will be 2 raised to power n, where n is equal to number of heterozygous forms, heterozygous genes, heterozygous conditions. If we see here heterozygous condition, this is one heterozygous condition, this is second heterozygous condition, this is third heterozygous condition. So our heterozygous conditions are 3, then it will have the number of gametes. It will be 2 raised to power 3, that is 2 into 2 into 2, it will be equal to 8. Now we will just determine these number of gametes manually by using Fock-Link method. By Fock-Link method, we can have the 8 different types of gametes. Let us see, suppose this is capital A, small a, capital B, small b, capital C, small c, it is crossed with capital A, small a, capital B, small b, capital C, small c. Now let us have the gametes. In gametes, what we have to see, first take these capital A and small a. Let me write first capital A here and small a here. Same capital A here, small a here. This capital A, this is crossed with this capital B, small b. Also this will be crossed with capital B, small b. So this K, capital B, small b. Also this small a, it is crossed with capital B, small b. And this small a, it is crossed with capital B, small b. Now remains here capital C, small c. This B will be crossed with capital C, small c, capital C, small c. Also here capital C, small c, capital C, small c. This will be capital C, small c, and this will be also capital C, small c. If we take the gametes for it, it will provide, this will provide us 4 gametes, this will provide us also 4 gametes. Like capital C, small c, capital C, small c. So let us have the gametes first. This will be 1, 2, 3, 4. 1, 2, 3, 4. So each parent will produce 8 different types of gametes. It will be capital A, capital B, capital C. Capital A, capital B, capital C. Now capital A, capital B, small c. Capital A, capital B, small c. This will be capital A, small b, capital C, capital A, small b, small c. These are the 4 different types of gametes. Now coming to here, it is small a, capital B, capital C. This will be small a, capital B, small c. Same, it will be small a, small b, capital C. This will be small a, small b, small c. So these are the 8 different types of gametes. So here 8 different types of gametes. Also here will be these 8 different types of gametes. It will be capital A, capital B, capital C. This will be capital A, capital B, small c. This will be capital A, small b, capital C. This will be capital A, small b, small c. Again here, it will be small a, capital B, capital C. This will be small a, capital B, small c. This will be small a, small b, capital C. This will be small a, small b, small c. Like this only. So these are the 8 different types of gametes. Now in order to cross these gametes, we need the help of Punnett square. So Punnett square is the square in which we can determine the different genotypes and different phenotypes. So let me rub this. So let us take the help of Punnett square. In Punnett square, we are having this table. And when the number of gametes is 8 from each parent, the number of zygotes will be 8 into 8, that will be 64. 8 into 8, that will be 64. So here 64 zygotes will be formed. Like this. This one be 1, 2, 3, 1, 2, 3, 4, 5, 6. Be 1, 2, 3, 4, 5, 6, 7, and 8. Here will be also 1, 2, 3, 4, 5, 6, 'So this will be 1, 2, 3, 4, 5. This will be 6. This will be 7 and 8. 7 and 8. And in this, we will have capital A, capital B, capital C. Then capital A, capital B, small c. Is this clear? Again, we will have capital A, small b, capital C. Capital A, small b, small c. Then we will have small a, capital B, capital C, small a, small b, capital B, small c. then we will have capital A, small b, capital C, small a, small b, small c. Is this clear? Now coming to, okay, now from here we'll have capital A, capital B, capital C, like this only. It will be capital A, capital B, small c. This will be capital A, small b, capital C, capital A, small b, small c. This will be now small a, capital B, capital C, small a, capital B, small c. This will be small a, small b, capital C, small a, small b, small c. Now we have to make a cross. We have to make a cross. Like we can see, this will be capital A, capital A, capital B, capital B, capital C, capital C. So all the alleles in this progeny, all alleles are dominant. So number of alleles is 6. Is this clear? Is this clear? Are you getting this point? This is a quantitative inheritance, and we are just counting the dominant alleles, that is capital alleles here. So capital A, these are the two alleles. Capital B, two alleles dominant. Capital C, alleles dominant. So total number of dominant alleles is 6. Now I will not make a cross, or I will not write these letters here. I will write only the dominant alleles here. So here are 6 dominant alleles. So in next column, what we will see, we will count only the number of dominant alleles. So here 3 dominant alleles, here only 2 dominant alleles. 3 plus 2 is equal to 5. I may write it here only 5. Not capital A, capital B, capital C, small c. No. We will count only the dominant alleles. So here are dominant alleles only 5. Is this clear? Coming to here. Here will be 3 dominant alleles and 1 dominant allele. This is but 1 capital C. Then it will be 5 dominant alleles. Here it will be 3 plus 1. It will be 4 dominant alleles. Here it will be 3. 3 plus 2 is equal to 5 dominant alleles. Also here will be 3. Here will be 2. This will be 4 dominant alleles. Here it will be 3 plus 1. It is 4 dominant alleles. Here it will be only 3 dominant alleles. If we see in this column, it will be here 3 dominant alleles. Here only 1 dominant allele. 3 plus 1 is equal to 4. So here are 4 dominant alleles. Other are recessive alleles. Here if we see 3 dominant alleles plus 1 and 2 dominant alleles. No, sorry, only 1 dominant allele. This is only C. Small a, small b, capital C. So 3 plus 1 is equal to 4. And here only 3 dominant alleles. These are 3 recessive alleles. 3 is here. Again coming to here. Here are 2 dominant alleles. Here are 3 dominant alleles. 3 plus 2 is equal to 5. Is this clear? Here are 2 dominant alleles. Here are 2 dominant alleles. It will be 4 dominant alleles. Here are 2. Here is 2. It will be 4 dominant alleles. Again here are 2 dominant alleles. Here is only 1 dominant allele. Then it will be 3 dominant alleles. Is this clear or not? Clear? Now coming to here are 2 dominant alleles. Here are 2 dominant alleles. It will be 4. Here are 2 dominant alleles, and here is only 1 dominant allele. It will be 3. Here are 2 dominant alleles. Here are 1 dominant allele. It will be again 3. Again here will be 2, and here will be no dominant allele. So it will be 2 here. Is this clear? Coming to next. Here will be the 2 dominant alleles plus 3. It will be 5 dominant alleles. 2 dominant alleles plus 2 dominant alleles. It will be 2 dominant alleles. Sorry, it will be 4. 2 plus 2 is equal to 4. Now, if we see here, 2 dominant alleles, it will be 4. Here will be 2 dominant alleles, only 1 dominant allele, it will be 3 dominant alleles. Here will be 2 dominant alleles, and here will be 2 dominant alleles, there will be 4. Sorry. Here will be 2 dominant alleles. Here will be only 1 dominant allele, then it will be 3 dominant alleles. Here will be 2 dominant alleles, and here are 1 only, so it will be 3 here. 2 dominant alleles, no dominant alleles, then it will be 2 here. Here we will see here is only 1 dominant allele, and here are 3 dominant alleles. 3 plus 1, this is 4. Is this clear? Next is, this is 1 dominant allele and this is 2 dominant alleles. Then it will be 3 dominant alleles. Here will be 1, here will be 2. This will be again 3 dominant alleles. Here will be 1 plus 1, it is 2 dominant alleles. 2 plus 1, 3 dominant alleles. 1 plus 1, 2 dominant alleles. 1 plus 1, 2 dominant alleles, and 0 plus 1 is only 1 dominant allele. Now coming to this column, here are 2, here are 3. 3 plus 2 is 5. Again 2 plus 2, this is 4. Now 2 plus 2 is 4 again. 2 plus 1 is 3. And then 2 plus 2 is, this is 4. Again 2 plus 1 is 3. Again 2 plus 1 is 3. This is 2 dominant alleles and here is no dominant allele, then it is only 2. Again we come here. This is 1 dominant allele and 3 dominant alleles, it is 4 dominant allele. This will be 3 dominant allele. This will be 1 plus 2, 3 dominant allele. This will be 1 plus 1, 2 dominant allele. This will be 1 plus 2, 3 dominant allele. This will be 1 plus 1, it is 2 dominant allele. This will be 1 dominant allele and 1 dominant allele, it will be 2. 1 dominant allele, 0, it will be 1. Here we see 1 dominant allele, 3 dominant allele, it will be 4. 1 dominant allele, 2 dominant alleles, it will be 3. 1 dominant allele, 2 dominant alleles, it will be 2. Here will be 1 dominant allele, here will be 1 dominant allele, it will again be 2. 1 dominant allele, 2 dominant alleles, it will be 3. 1 dominant allele and 1 dominant allele, it will be 2. 1 dominant allele and 1 dominant allele, it will be 2. 1 dominant allele, 0 dominant allele, it will be 1. Is this clear? Coming to here, 0 and 3. 3 dominant alleles, 0 dominant alleles. Now, this is all 0. We will consider these things only. So, this will be 3, this will be 2, this will be again 2, this will be 1, this will be 2, this will be 1, 1 and 0. So, these are the number of dominant alleles in the progeny. If we see the number of the dominant alleles in the progeny, we will have some graph here. The data we collect from this Punnett scale is like we have number of phenotypes 7. Let me have it. Capital A, capital A, capital B, capital B, capital C, it is having 6 dominant alleles. Is this clear? Now, capital A, small a, capital B, small b, capital C, it is having 3 dominant alleles. And small a, small a, small b, small b, small c, it is having 0 dominant allele. So, in this 3 dominant alleles, if we see this is capital A, small a, capital B, capital C, it is having 5 dominant alleles. Capital A, small a, capital B, small b, capital C, it is having 4 dominant alleles. Now, coming here, small a, capital B, small b, capital C, it is having 2 dominant alleles. Small a, small b, capital C, it is having 1 dominant allele, and this is having 1 phenotype, 2, 3, 4, 5, 6, 7. These are 7 phenotypes and genotypes. Is this clear? So, let us count how many. This will be called as very dark. This is the dark color. This is light dark color. This is intermediate. This will be light color. This will be very light color. And this will be white. Yeah, mulatto. This is white color. So in this color we have so many different traits. If we count the number of individuals here with 6 dominant allele, we will find only 1. So this number 6, it is counted, so that I am making this circle here. The individual which having all the 6 alleles dominant, it is only 1 in number, so let me write it 1 here. Second one, the 5. How many 5 dominant alleles or organisms we will find? 1, 2, 3, 4, 5 and 6. This will be 6. So let me write it here. It is 6. Coming to these 4 alleles. This is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14 and 15. So number of individuals which are having 4 dominant alleles is 15. Now coming to 3 alleles. If we see 3 alleles, this will be 1, this will be 2, this will be 3, this will be 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20 आना चाहिए. It should be 20. Somewhere I have made a mistake, but it is 20. So here we see this is the 3 dominant alleles, 17 number. This will be 18 number. This is 19 and this is 20. So the individual with only 3 dominant alleles, it will be 20. There are 20 individuals. Now coming to here, this will be repeated. There are 2 dominant alleles, it will be repeated. This should be 15. This one, it should be 15. So let us count these 2 dominant alleles. Suppose, where is the first 2? It is 1, 2, 3, 4, 5. This will be 6. This will be 7, 8, 9, 10, 11, 12, 13, 14 and this is 15. So this will be 15 here. Now if we see with 1 allele dominant, it will be 6. 6. So let us count 1. This will be 1. This will be 2, 3, 4, 5 and 6. So there are 6 individuals with only 1 dominant allele. And if we see with 0 dominant allele, it is only 1 here. So it is with 0 dominant allele, it will be 1 only. Is this clear? Now if you count these 6 plus 1, 7, 7 plus 15, it will be 22. This will be plus 20, it is 42. 42 plus 10, 52, 57, 50. कितना हो गया ये? Okay. So if we see that very dark color is having only 1 individual with 6 alleles dominant, 6 individuals with 5 dominant alleles, 15 individuals with 4 dominant alleles, 20 individuals with intermediate color or we can say 3 dominant alleles, and 15 individuals will be of 2 dominant alleles, 6 individuals will be having only 1 dominant allele, and one individual will be with zero dominant allele. If we count these figures, you will get total number of individuals 64. So, if we take a graph of this, in this graph, we will be having a graph like parabola. This is here number of dominant alleles. So if we see this, the individual with 6 dominant alleles is only 1. Suppose it's the number of 1, 2, 3, 4, 5, 6. So it will be 2, 3, 4, 5, 6. And here are the number of dominant alleles 1, 2, 3, 4, 5, 6. So in the first individual, we are having only one individual which are having 6 dominant alleles. So our graph will be like that. This will be one individual with only one dominant allele. Sorry, with 6 dominant alleles is only one individual. If we have the second one with 5 dominant alleles, then we will have 6 individuals. Sorry, only 6. Sir, usko pass kijiye. So from this data, we are getting these genotypes, and in these genotypes, we are having these number of dominant alleles. Here, the 6 dominant alleles are here, 5 dominant alleles are here, 4 dominant alleles are here, 3 dominant alleles are here, 2 dominant alleles are here, 1 dominant allele, and 0 dominant allele. Now we will count the number of the individuals with 6 dominant alleles. It is only 1 here. Is this clear? Second, the number of individuals with 5 dominant alleles is only 6. And number of individuals with 4 dominant alleles is 15. 3 dominant alleles with 20. 2 dominant alleles, 15 individuals are there. 1 dominant allele, 6 individuals are there. 0 dominant allele, only 1. If we count the number of odd individuals, we will get 64. So we are having 64 progeny, or we can say 64 zygote. If we take a graph of these 64 individuals with x-axis and with y-axis, in the x-axis we will have number of dominant alleles. It's increasing from this point to this point. It will be increasing. So let me take an arrow like this. It is the direction of increasing in these dominant alleles, and this will be taken against the number of individuals, and number of individuals will be increasing like this. Is this clear? So if we see the first thing with dominant allele 1, this number of individuals, the number of individuals we will see with 1 dominant allele, I think this